So this is a little outdated, and I am from Indiana, and Indiana, most of the other stuff other than sampling was in Indiana, so I'm heavily Indiana based. But as probably most of you in this room know, there's actually really a pretty large struggle for the conservation of herpetofauna throughout the nation. So this is our list on our website that is still unfortunately outdated, but it is the most recent one we have on the DNR's website. And pretty much many species of amphibians and reptiles are in decline. And in Indiana alone, we have 12 amphibians and 19 reptiles listed in our state of either special concern or state endangered. And some of those do carry over to the federally threatened side, like the Massasauga. And conservation of many of these species relies heavily on the conservation of the site. Protecting the habitat is the biggest thing that we can do as conservationists. But part of that issue with a lot of reptiles is there's a large struggle to accurately describe their populations, their numbers, and where they're even located. So, as I already mentioned, Massasaugas are no exception to the rule. So this was a map from a study done in Ontario back in 2016. The gray areas that I'm hoping you can kind of see are the historic distribution of eastern Massasaugas, and the blue is the current distribution. And as you can see, pretty much most places are decreased in locations by half, or if you're Minnesota, even more than half. Pretty much the only relative stronghold is Michigan. So that is why Michigan was my place of study. So there's a lot of factors contributing to the decline. Like I said, I'm probably preaching mainly to the choir here, but for those that may not know, one of the biggest things is draining of Massasauga habitat. That can include wetlands, prairies, marshes, either for development, for agricultural usage, various types. Road construction is a really big issue. Road construction is kind of a double-edged sword, though. It's not just the fact that the roads are there. It contributes directly, people accidentally running over snakes that are there. But it's also indirect because you're actually physically separating out different populations. And if we go back to this one, you can kind of see, you can see it well, but there's not a lot of continuity in some of these peripheral type of populations. So it's kind of important that we try and keep populations together. And then further for roads, there's been a lot of studies done saying that snakes don't like to cross some types of substrates, or they're not willing to travel as long of distances to find mates and find other species. And then a big one is still human persecution. A lot of people still have, unfortunately, a view of all snakes are evil, and all snakes of any type, regardless if they're venomous or not, should be killed on site. So that's a really big issue, and if you're a rattlesnake species, well then you're just kind of doubly doomed. So why does accurate detection matter, which is the crux of why you use eDNA research? A lot of the issue is that there are techniques that exist to facilitate the recovery and rely on Massasauga ecology. However, we don't really understand really well overwintering site selection by many snake species. We also don't fully understand all the spatial requirements, or what we do have is maybe very specific to a certain area and doesn't transcend everywhere. And then many eDNA studies look at the efficacy of testing eDNA, but we don't look a lot at what that means. So you have a test, but what do the environmental effects have on that eDNA? How does it change over time? So this kind of helps bolster the arguments that we wanted to argue to test and see how eDNA can be used for Massasaugas. And this is Pierce Cedar Creek, if you're wondering. So like I said, many of you in the room probably know a lot of the stuff that I'm covering at the introduction, but traditional survey methods for snake species is typically a visual encounter survey. And that's what we use to estimate diversity and abundance of species. But pretty much what that means is if you see it, you count that as a detection. If you don't see it, that's a non-detection. But the problem with the non-detections is it might have been there, you just might not have seen it. Or maybe you're being lazy that day, I don't know, or it could actually have not been there. So it's really kind of hard to tease out the two separate reasons of why there's non-detection. And it's kind of further exacerbated for species like Mastosagus when they're cryptic. They have the camouflage, they're hard to see, and then as a whole, not just Mastosagus, but snakes being ectotherms, they actually are only active during certain times of this year, certain times of the day, so it gets really hard. If you're not using traditional survey methods, if you're trapping, there's a lot of stress during handling and capturing. It can be expensive, not just in your equipment, but the number of hours that you have to have people out there actually looking for them. And then if you've heard about snake fungal disease, or what is commonly known as snake fungal disease, then you have to worry about a whole other issue of spreading that disease as you travel to different sites. So for those of you, I'm guessing most of you are going to see this, but how many of you can instantly spot the Mastosaga? Okay, so most of you I'm guessing because you're here for a reason, because you haven't. But it's really not always that easy. And especially if you're out walking around and you're not thinking, that spot is where I should be looking right now. I wasn't looking at this spot, I was testing a burrow and reached my hand down and happened to spot it on Halloween, which is kind of fun, but. So basically it all comes down, there's gotta be a better way, right? It's not that easy just to find all the cryptic species all the time. So that's where environmental DNA or eDNA comes into play. It's kind of like doing forensic lab work, except instead of people, it's for wildlife. The DNA from an animal is found in soil and water in the environment. There has been a study that they've done it where they've used air somehow, but I really don't fully understand that mechanism. But essentially, the DNA starts accumulating through things like shed skin, nitrogenous waste, fecal waste, reproductive products, skin, fur, what have you. It's been gaining popularity over the last probably 10 to 15 years. There's a lot of benefits that are thought to be associated with eDNA, which is why it's becoming more and more popular. So some of the benefits of eDNA, one of the biggest ones is it limits stress to the animals while you're handling them. I said, snakes there's a whole variety of animals that stress at different levels, but either way, there's still going to be stress associated with being captured, being handled. It also helps if you're just testing on one spot instead of traipsing around. Hopefully it would limit your spread of disease if there is a disease there. Rapid detection, if you have a protocol in place, ideally you could take a sample and filter it within 24 hours, know yes or no, did you have a positive hit of a species being in a location. And then finally, once it's developed, overall there is a lot of monetary savings associated with it. And then we already have some poster children for eDNA, eastern hellbenders, longfin pilot whales, Burmese pythons in the Everglades, big-headed and silver carp, which are the two Asian carp species. I'm sure they'll throw into the black carp coming up. But it's already used for a lot of things, so there's reason to believe that we can use it for other animals. So they're using it for hellbenders, which are just super rare, but we're also thinking about using it for invasives, because as you also probably know, if you can detect an invasive right away, that's your best chance of eliminating before it becomes so large that you can't deal with it. So with all that being said, we decided that Mastosagas are probably a really good candidate for eDNA surveying. That is because specifically in Michigan, there's documentation where they do overwinter in crayfish burrows. They overwinter in these crayfish burrows because it has water below the frost line, and that water insulates them during the winter, keeps them from drying out, from when it's freezing. Sorry guys, I was talking a lot yesterday too. So. And they also show fidelity to their overwintering sites, so they keep coming back. And then for another added degree of the preservation of DNA, if you think about a burrow, hopefully it's cool, it's dark, and hopefully that DNA is going to be well preserved and collected and concentrated at the bottom of a crayfish burrow. So that's why we figured we'd try that. And then again, Michigan does have multiple primary crayfish burrowing species where they actually, instead of spending all the time in the water, they actually spend most of their time in the burrow. So So my research had two main objectives. One was to develop and test an eDNA assay to identify Eastern Massasaugas. And then the second one was to try and take abiotic characteristics at those burrows to see if we could correlate to see maybe what characteristics of those burrows maybe correlate with a higher detection rate in a burrow or more DNA or something along those lines. So this is kind of the overview of my project. It's a really confusing process, so I'm hoping showing this to you at the beginning will kind of help you think about all the moving parts. So the first part is I had samples in the lab and in the field as a control, and then for the actual study to see what's happening. So once you have your samples, you do have to filter and extract them. And then you have to do the results in the analysis. But to do the qPCR, you actually have to develop your primers and your probe, which is part of that. And then once you have your positive or negative detections, you can put that into, I use occupancy modeling to try and decide where we were going with that and what it actually meant. So I'm going to start with the collections first. So the first thing that we did is I actually used laboratory house Massasaugas to collect some controls. So these snakes were actually brought to Purdue, Fort Wayne, for a different study in a different lab where they had transmitters planted in. They're from Grayling, Michigan snakes, so that's part of that research project up there. But before they were put into these tubs, what I did is I cleaned these tubs, bleached them, let them dry thoroughly, set them up so they're appropriate for snake species. And then I took distilled water and then autoclaved that and put it in there to make sure we had super clean water. And I tried to just put a little bit, enough for the snake to swim around, but not for it to be totally submerged. And then I took samples at days 0, 1, 2, 7, 14, 28, 56 days later. My day 0 is actually the day that the snake came out, and I did take a sample before the snakes even went in. So the idea of this is kind of to see, does it work in a lab setting, but then to also to see, does the DNA in fact degrade over time. So then, you're probably going to have a hard time seeing it, all the samples in the lab were treated the same way as the water samples from the field. The only thing that I didn't have in the lab was a sediment sample, which we will get into. So my field site was here at Cedar Creek Institute. The first site, Cedar Creek Institute in Michigan. Like I said, Michigan's this relative stronghold for the species, but we've actually had studies of various types of Massasaugas at this location since I think 2004. And we also, by me talking with some other researchers out here, there's varying levels of occurrence of snakes in these different areas. So that's why we try to see like an array of usage for the areas. So the first one is actually right outside the research lab. Here's the one hill, and then across the street on this side. So that was the first area where word of mouth and other reports, snakes are usually there. The second area is actually this wooded area between the two sites, where to me I was told that they have been spotted there, maybe slightly less frequently than the first site, but they have been spotted out there pretty regularly. The third site is actually back behind the research center, where they have been spotted, but because it's more like a mucky area, it's likely way too wet for them to actually overwinter back there. So ideally they'd be overwintering here, probably there, but not necessarily for overwintering here. And then the last site is actually on the edge, Cedar Creek's edge of Armon Lake, at the newly acquired part of the property, or relatively newly acquired, where from our records and from past research, they have had no reports of it at all. So that's why we picked those areas. And so these are all within what we would consider a snake's home range. So it's not completely off the wall, far distances either. So I started sampling in May. And we try to do May because that's when they're starting to emerge. So we wanted to try and correlate that if they're going to be out of the burrow, so I'm not disturbing them during their overwintering time. But as they're emerging, so hopefully any DNA that we did get that was positive can be said, well, it's probably because the snake was recently there. I did 15 rows at each site, so I had a total of 60 different sites. And then pretty much I rigged up, with the help of my husband, this hand pump to pull water out. It's essentially just a syringe with a catheter adapter with some sterile tubing put over the end, and just fed it down the hole and pulled it up. And so the first thing I did at every site was to pull up the water, put it in a container, wipe off that container on the outside, and then put it in my cooler. And that's really important because eDNA is super sensitive and easily contaminated. So even before I went out to the field, you always have to have controls, right? So before I even got to this site at my house, I'm pouring distilled water into a tube as a sample, wiping it down and putting it in the cooler on the same ice. So that way we can check for contamination at all points. So that's what I did at every site. After I collected the water, because that was the first thing, because we wanted that to be the most non-contaminated, I drew up additional water and used a YSI meter to test the pH, temperature, and dissolved oxygen to see if those were the environmental factors maybe affecting where the snakes are going. And then we also took some environmental measurements, like burrow diameter, distance to different types of cover. It was like log, shrub, tree, water. And then later, after I had all my points, I actually used an aerial map, an ARC map, and ESRI to try and determine the distance to the nearest path. For all of the cover habitats, anything greater than 20 meters, it just got left as greater than 20 meters. The pathway, I actually tried to go to the nearest kilometer, whether or not it was super far or super close. And again, sorry, I should have done this earlier, but the pH, temperature, and dissolved oxygen, they all have varying effects of preserving DNA. If it's over-oxygenated, that actually degrades your DNA faster, but higher temperatures degrade, but you also want, so I don't tell you acidic conditions actually degrade, so you're looking for more basic types of soil. So those are all coming to factor of how eDNA behaves in the terrestrial setting. So after I collected everything, put it in the cooler, every day I drove it all the way back to Fort Wayne and I filtered it. You always want to make sure you filter within 24 hours, so I kept it on ice. If I couldn't do it that night, I just put it in the fridge, in the ice, and I filtered it the next morning. So essentially, as long as it was filtered within 24 hours. Because we were trying to get more data from the limited sources we had, we actually did try and separate a sediment and a water sample. So prior to me filtering and extracting, I actually spun the samples that I had from the field, and I just decanted the water into a filter and used a peristaltic pump to just suck it through, and ideally the DNA would get caught on those filters. And then what I did was I put those filters in with CTAB and PVP. CTAB helps with the lysis of the cells to expose the DNA, but also helps to preserve it. And the PVP helps to absorb phenols that are found in plant matter, so that way you don't have inhibition in your PCR later. The only difference is for the sediment samples, since there's not a filter there, I actually added just a different combination of the CTAB and PVP directly to the leftover sediment in the tube. And I actually left it in the lab at room temperature for 2 weeks until extraction. There was a paper that said leaving it for some amount of time in CTAB actually increased their detection rates. 2 weeks was probably the maximum. Don't ask me why we decided 2 weeks at the time, but that's what we went with. So they sat for 2 weeks before we did the rest of the extraction. So we did a phenol extraction, which is allegedly better for getting your samples from the environment. So day one, you did the extraction, left it in the freezer overnight, and the second day is when you extracted your pellet, let it dry, resuspended it in buffer, and it sat at negative 80 degrees until I was ready to do the qPCR stuff. All right, that was the easy stuff. So the next part is, how are we going to detect the DNA if we have it? So the answer that most people use when using eDNA is what's called real-time quantitative PCR, or qPCR. And basically what happens is you amplify your target DNA, at the same time you're also amplifying a probe, okay? That probe is within your strand of DNA, so like pretend my finger is a strand and the probe sits somewhere in the middle, okay? And then on either side of that probe there's a reporter and a quencher. So every time you run a cycle, the polymerase of qPCR breaks that apart and it emits an excited fluorophore. And that's what gets measured to see comparatively how much DNA you have. And so theoretically, if you have more DNA, you should have a higher reading of the fluorescence in the machine. So the fluorescence is measured at the end of each cycle of PCR. And you also put in standard curves to compare the known amounts against, and you can use the standard curve with the measured fluorophore to try and see about how much DNA you had in your starting material. So the benefits of this, it allows you to actually estimate the amount of DNA you had, and then the probe, in addition to your primers, adds extra specificity to the DNA that you're trying to extract. So it's just another level of saying this is in fact the DNA we wanted and not some other random DNA. So to do all that, you have to design good primers and probes. We use the mitochondrial Co1 gene. It has low within variation for Mastosagus, but it's actually high variation to other snake species, and it's actually becoming more commonly used for markers for speciation of snake species. These were all lined, and we use the BOLD and GenBank databases, and so there's a lot of the computer work too for whatever extensions I could find. I just put them in there for any snake species that I thought might be in conflict with my Mastosaga DNA. And in those sequences, you're looking for somewhere between 75 to 200 base pair segment. And then for your primers, you want to try and have at least one or more mismatch in the forward and reverse primers. So for me, I had 2 base pairs that were different in the forward primer and one in the reverse. These are all the snake species that I used at the time. Mastosagas, blue racers, brown snakes, ribbon snakes. I said it's probably not by no means comprehensive, but I felt it served well for the Michigan, Indiana area. And once you have your primers designed, you design your probe, and that is actually much shorter, so it's only about 20 base pairs, that's what you're shooting for. And that sits within your smaller segment of DNA. And then using different things you can check for heterodimers, homodimers, hairpin formation. So essentially, we ran another tool to check to make sure that any DNA that we did get was not because the primers are just forming to each other. So then I ended up with a 21 base pair each forward and reverse primer, and then 24 base pair probe. And so to make extra sure, before I even did probing, I actually just ran regular PCR with my primers and ran with the snake species I had available. I had others, but I just could not get the DNA to become extracted. So I had some things like the ribbon snake, the hognose snake. Those were all there as well, but I also had some off-the-wall things like a red belly snake that's probably not as likely. So we tried to just think of any DNA that we had that was snake related, and we threw it into the mix to make sure. So as you can see, there's a lot of DNA. That's my Mastosaga, and that's the 152 base pairs. So that was what we did to specificity. And again, I shouldn't go back so much, but to specify how sensitive eDNA is contaminated, my extractions and my qPCR and my setup, I had 3 different labs working. Because once you extract it, you take it somewhere else, and once you've extracted that and run your PCR, you go somewhere else. So it's just, you need a lot of lab space, just a disclaimer. So then, once you have your primers, this is kind of what your readout would look like if you did a qPCR. Again, these are the excited fluorophores, these are the readings that you get, this is the background noise. So for my stuff, the blue, it indicates that it's Mastosaga DNA. The green is actually what's called an internal positive control. And this is just there, you want to see it amplified no matter what, as to know that you're not being inhibited by anything. So if you have your negative control, it will not have a blue mark, but it should always have a green one. And if there is inhibition, then, or if you don't have a green signal, that's indication that you should probably go back and clean your sample because something's probably going on that's inhibiting PCR. So those red lines all indicate the standard curves, and I know it's really hard to see, but you can kind of see the stair-step formation. So this was my highest amount of DNA, of known DNA. down to the lowest, so more DNA shows up quicker with a higher amplification. The black, excuse me, it's a positive control, so that's a known Massasauga DNA that I put in there that I know for a fact that it should amplify. The orange is an unknown sample, so this was like all my field samples and lab samples. I don't know what's going to happen with this one, put it in there. And the green, again, is the IPC. And up here you can see, I've highlighted this one because you have this green emission, but then down here you have a blue emission way past 40 cycles, which could just be the noise at the end of so many cycles. And ideally, there's nothing up here, so that indicates that it's not inhibited and it is a true negative. So once we did all this, some of my results. So the biggest thing with eDNA for reporting purposes and to see if we can even use it regularly in wildlife and testing is kind of know how sensitive it is to see if you're actually working. In the perfect world, you should be able to detect 3 copies of DNA per milliliter. As you can see, mine is not that low. We kind of did a really rugged test for this and just did a theoretical limit of detection by using a standard line, and we came up with, I could detect about 49 copies. That's the lowest that I could get my samples to go. We did also send out 10 of our 12 samples for Sanger sequencing. We sent out 12 samples for sequencing. 10 of them came back positive for the targeted Massasauga DNA. 2 of them just didn't work. So I don't know if they're negative or positive, they just didn't work. So then some other results for the laboratory house Massasaugas. I did have detection in 11 of my 21 samples. I had 3 snakes. Each dot is an average of the replicates that I took from that day. What you can see is, so the first snake, it starts decreasing, but then there's this increase and then another decrease, and it's kind of similar for day 2. What I did notice for this that was interesting is that both of these snakes actually defecated in my container. And this decrease, or this spike actually starts happening about when I have it noted in my notebook that the fecal matter was completely broken down and just disseminated throughout the water. The third snake did not. I had pretty almost clean water after I took it. I know it's not clean, but it was clean looking water. And after 2 days, I couldn't detect the DNA in there. So the CQ values are from 19 to 40 cycles, so that means I averaged anywhere from detecting 2 copies all the way up to 9.9 times 10 to the 5 copies. So I still did get quite a bit of reading of DNA in my lab house, Massasaugas. For the eDNA field samples, this was super interesting. So on your left was the water, and on your right is the sediment samples. In the water samples, I had 6 positive detections at the burrow level, but 9 of my replicates. So each sample you take, you're actually running in a triplicate. What was really interesting is we had the most hits at the area 1 where we know that they are occurring, and there was not a lot at area 4, which is exactly what we expected. However, when you look at your sediment samples, I actually had no positive hits where we know Massasaugas are hanging out every day. And I actually had my strongest hit overall came from a sediment sample taken out by our own lake where we have no documentation of where they have been occurring. So that's what I got from my field samples. And then to try and talk about the different sites, I used ANOVA and MANOVA to site comparison just to see, are these sites even actually different when I look at the temperature, the dissolved oxygen, are they even actually different? So MANOVA pretty much indicates that there is actual statistically significant difference at all the sites. So I try to see which ones were more similar using Tukey HSD. If you put in Turkey, R doesn't let it work. It's super fun. So what we actually found out for which sites are most similar, area 1 and 2, which are the areas where we're most likely to see snakes, were actually most often the most different from each other. The only thing that they really had in common was the relative distance to the nearest pathway. And area 1 and 4, whereas actually most similar among all the sites, except for the distance to nearest pathway. So it's kind of interesting that all the other factors contributing made 1 and 4 more similar.
Can you just remind us what area 1 and 2 is?
Sorry, yes. Area 1 was the one right outside the lab where we know they're happening. Area 2 was across the way in the forested-ish upland area. Area 3 is back way behind the lab, and then Area 4 is back at our lake.
Did you check the pathway too for DNA? Or you just assuming the snakes used that path that was nearby?
No, I just any pathway that was near, so it could be a footpath. It didn't matter. Any path that was pretty much designed for human passage. So it could have been a road, it could have been a mob trail. So once we had all this information, we used eDNA occupancy to try and model what was happening. So occupancy is better than presence absence in the way that it, as we mentioned before, absence can't tell you, was it not really there or did you just miss it? Occupancy modeling actually takes this into consideration. And it uses Bayesian technology to pretty much with your covariates to see, so I have different rows, right? Is there any level of covariate? So if I change the pH, does that change my likelihood of finding a positive detection? If I change the temperature, does that change it? So when you run the model and it runs the Bayesian system, it actually shows you the model criterion data, so which one you should select. So you try and find if any of these covariates actually change. And the way the Bayesian level works is like a hierarchy, right? So you have the occupancy at a location, so my area is still around our lake, or at the, yeah, I'm sorry guys, it's a struggle, but, so if it's at an area, what is the probability of being in that area? And then if it is at an area, what is the probability of you finding it in a burrow? And if it is in a burrow, what is the probability of you finding it in a qPCR replicate? So that's how it kind of works to sample out your occupancy modeling. So it generates something similar to this, and again, you're just looking at the criterion selection modeling. So for my water samples, one of them was best if you didn't have any covariates, so it didn't really matter. But the other one showed that the overall DNA concentration, which if you have a higher DNA concentration, you're likely to have a positive detection, which is pretty intuitive. And you can see that here. However, when you ran the sediments, there's actually no indication of the change in temperature, change in pH, change in DO that mattered, which is likely due to the fact that I only had 60 samples total, and 15 at each site. So to recap for my objectives, to develop and test an assay, and then to correlate abiotic characteristics of those rows to see if it mattered in site selection. So again, I detected 49 copies. There's another student that she also did Massasaugas in Illinois. Her paper says that she got down to 4 molecules, and then other snake papers have gotten down to 0.36 molecules of DNA. So mine's definitely a lot higher than those. Like I said, the magic number kind of seems to be 3. I definitely wasn't at 3. And like I said, 10 out of 12 samples did come back positive for the eDNA that I ran came back and tested positive for Massasauga DNA. eDNA can be detected in a water setting 56 days after a snake has been removed in a lab setting. I do have reason to believe that broken down evenly distributed matter actually helps you benefit with your detection. And there's also a decline in the estimated starting quantity of your DNA over time, which is what you would expect. So it helps you kind of maybe think there can be a timeline possibly assigned in the real world. And then for burrow characteristics, all measurements contributed to the fact that I did have 4 different sites. The problem was areas 1 and 4 were actually much more similar than expected, and areas 1 and 2 were much more different than expected, with exception to the pathway. I maintain that there might be something along with traveling and routes and something. I've been told repeatedly that that's not the case, but someone else found something similar in a different type of study with Massasaugas. And then DNA concentration may affect your detection when using water samples, which makes sense. So what does that actually mean for the future? We're kind of thinking eDNA might not actually be that advantageous for snake detection. Rick Urach did it for Kirtland snakes, and in 100 samples, he got 1 positive detection. Baker, for her Massasaugas, she only got 2 in 100 samples. So I had detection at 6 different burrow sites. but if I actually use the rule of I can only positively detect down to 49 copies, I only had 2 out of 60 positive detections. I think we do need more understanding of snake behavior and ecology requirements. What are the snakes doing in their burrows? Are they defecating? Are they even moving around that much? Are they actually sloughing off skin cells like we thought? I mean, if you think about hellbenders, they're called snot otters. So think about what they're putting into the water for their eDNA detection. Think about Asian carp and fish, and think about how massive a Burmese python is and what's going to happen when they release their cells into the environment. So we might not just know enough. Maybe we should think about sampling during the actual overwintering site. There's definitely obviously a lot of concerns for that. We also, water tables changing in the burrows, that was definitely a factor when we were sampling. There are some burrows that I couldn't get my tube deep enough to actually hit water. There are some days that I'd go out there and I couldn't see water, and there's some days that there's water even with the opening of the burrow. So how is that affecting eDNA concentration? So the positives that I have is that I did try and follow all the reports and requirements for eDNA reporting forms. I tried to make sure I knew my limit of detection. I tried to make sure I had negative controls. I tried to make replicates. So if nothing else, at least it's groundwork for maybe future eDNA studies to build off of, and it's just another species that has been used and tested with an assay for eDNA. I definitely, before I get to questions, I want to thank everyone that was out there. Indiana Academy of Science did help fund this. I had a really great group of advisors at Purdue, Dr. Jordan, Dr. Kingsbury, Dr. Soul. Pierce Cedar Creek Institute was pretty awesome. Sarah hooked me up with a lot of the stuff that I needed. AJ literally drove his cart for me so I didn't have to haul everything so far. Dr. Jen Moore walked with me on sites, and she was out there the first day troubleshooting with me just to see if we could sample the burrow how we wanted to. And then her graduate student, Dana Bradkey, actually at the time came out and she walked other sites with me to see where she thought might be a good idea for the areas of sampling. The grayling crew are all the people that talk to me about husbandry of keeping snakes in water overnight. They're the ones that are doing all the transmitter work up in Grayling, and they were kind enough to drive all the way down from Grayling to Purdue. They had to do it anyway, but they were kind enough to put off their research so that I could actually have them sit in water overnight. Sample gathering crew, one of them is actually in the crowd. I made Jim come out with me and collect samples with me. Your picture was on here, I'm sorry, but I didn't have a picture of you. But he did come out and help me. I dragged him through the woods too. Some graduate students, and your family is always really good for free labor. So I got my father, my husband, my father-in-law all out there. So with that, I guess, is there any questions?
You had found, I guess, I saw with the snakes in the lab that if they defecated, it seemed that you had a longer signal. Do we know if Eastern Mississauga rattlesnakes defecate in their holes?
No. Well, I don't. At the time, we don't know. So I think that was part of the issue is, are they defecating in crayfish burrows? So that's part of the struggle.
Do you know how often they poop generally?
It's probably not that frequent.
They do a lot.
Yeah, I figured out that.
So do you have any idea what, so you found 56 days is your time until you start seeing decrease in the lab. Do you have any idea what that would be like in the field? Because keeping them in a box with DI water is probably not the same thing as keeping them in a crayfish burrow in an open environment.
No, and that's what I was trying to hoping to get with the occupancy modeling. I just really don't think I had 4 sites and 15 samples. I don't think I had the numbers to actually get a better idea. There's other research that says that you can detect it 60 days in the environment after the fact. And now, after all this, I'm kind of wondering, I don't know, because a lot of the studies with eDNA is heavily associated with water, but there's really not a lot of the understanding of terrestrial environments. There is with the sediments, but they're starting to, the things that I read, they're starting to say like sediment holds it longer. So if I had a positive detection in sediment, that could be from a really long time ago, and there's probably still not been snakes in that area. That's a good possibility. So I think there's just too many unknowns. I really don't know how well it would correlate lake yet. I think that's the biggest question.
When you're sampling your crayfish burrows, are these burrows that we've established that snakes have lived in before, or is it just here's a crayfish burrow sample?
That's a good question. It was just a here's a crayfish burrow sample. I just kind of walked around and we were trying to do like a grid system to make it like random, but then it kind of ends up being random if you're walking around in tall grass anyway. So we just kind of came on the sites that we could find and just sampled what we could find.
Are these crayfish burrows just like straight holes in the ground, or do they connect underground when they're nearer to each other?
It depends on which crayfish made the burrow. That was actually going to be part of the study. I, for 2 months, tried to trap the crayfish, and I caught a frog. And I totally didn't know this until I started this research, but there's different levels of burrowing for crayfish. Some are primary burrowers, all the way down to tertiary, where they don't burrow as much. And depending on the species, they can get pretty elaborate in their burrows underneath. There was at one point trying to be a camera submitted down so we could like see a borescope so we could see, and we could see nothing but murky water. So it depends. That's what we wanted to look into, but we didn't get to.
This may seem like a dumb question, but how can you tell if a crayfish burrow? I mean, do they come out? You see it?
I can show you a picture of them. I never, to be honest, I never noticed a crayfish burrow until someone pointed them out to me, and now I see them everywhere.
Wow.
See if I know the picture. No, so for the ones that I picked, definitely not a stupid question though. You usually can see that they flung mud out. Some of them do have like they look like little balls of mud stacked on top of each other. I don't know how else to do. Some of them are capped. Some of them have been flattened out. So if I couldn't tell what it was, I missed it as well. But I was usually looking for signs of ones that were, and that could have been actually a much better study because you don't want to be overwintering in an active crayfish burrow because then the crayfish is just going to snack on you all winter. So maybe looking at activity of the burrows would also probably help to really try and understand some of that. Did you have a question?
No, I kind of answered.
Okay.
So you never actually saw any crayfish at all?
I saw crayfish, I just didn't see them in my traps.
So what's your thought? So I know you used the qPCR assay. What's your thoughts on just standard PCR for occupancy and detection probability type things? Why can't we just run standard PCR in a gel as compared to doing a qPCR?
I think you can. I think for us, we were just kind of following what people had been doing in the protocols, and then I think that probe and that added level of specificity just, I think it felt my advisor and I both kind of felt that just added another piece of mind of being like, no, this is definitely what we want it to be. I think there's another lab that I actually think sometimes he just does the PCR. And I mean, he's doing that full time, so he must have a system that does work. Thanks, guys. You've been an awesome audience.